Okay, hi everyone. Good morning. Happy rainy Monday. All right. Uh, so I had some trouble uploading the lecture slides onto Laulima. I don't know if Laulima is having problems or not. I couldn't do it last night, and then this morning the Laulima site was still having trouble. So I will try to get these slides up on the Laulima sites, later this afternoon. Okay, so keep that in mind. So this week we have a quiz that's due on Wednesday, like at normal time. So before Wednesday's class, that quiz will cover material that we will wrap up with today. So look for the quiz posted later this afternoon. And then I'll also get the study guide and hopefully the lecture slides up there as well. And then you will have a homework assignment that's due Friday night. Right, Friday night by whatever it is, 11:55, however Laulima kind of makes me have to set it. So look for that as well. Okay, all right. Let's go ahead and finish talking about some of the functions of P53, and in particular, how apoptosis is regulated inside of our cells. So this is a kind of a review to start off with, but it's good because let's make sure we're all on the same page of what we've been talking about. So we said that P53 is a very important tumor suppressor protein. So therefore, it's an important tumor suppressor gene. And P53 functions to decide whether the cell cycle should be permitted to continue forward if, for example, there's been some problem that's been encountered. The most obvious problems that people usually think of in terms of P53 is if there is damage to the genome, right? Either just due to the cell being exposed to some strong mutagen like radiation that has damaged the DNA, or if during S phase, during replication of the genome, there is some gross damage that was detected, right? Or even during things like mitosis, if things like the sister chromatids are not segregating properly. Right? So in any of those types of situations where you have big chunks of chromosomes maybe missing, deleted, right, or otherwise not segregating properly, the cell may not be able to fix that, right? So in those cases, P53 not only stops the cell cycle from going forward, right, but may need to signal the cell to undergo apoptosis. We studied how P53 functions as a tetramer, 4 molecules of P53 working together. And we said that the way our cells normally control the levels of P53 activity in it is by producing P53, but then pretty quickly getting rid of the P53. And we said that P53 is a target for ubiquitination by this MDM2 protein. Right? So when MDM2 associates with P53, remember they bind together at the sort of N-terminal transactivation domain of P53. There was that region I showed you last time, a really cool image of P53. Its shape kind of bound to the shape of MDM2 in that region. Right? Then the MDM2 will go ahead and transfer ubiquitin groups onto the P53, resulting in these ubiquitin sort of flags, so that the P53 then gets moved out of the nucleus into the cytoplasm where it gets recycled by the proteasome. Right? That's how normally cellular levels of P53 are kept quite low. Right? However, so let's look at this, right? So how is this controlled and influenced? So the MDM2, right, is originally expressed by the MDM2 gene here, right, which is controlled by certain mitogenic and cell survival signals. So in this particular case, if you have ETS factors, so ETS are a family of proto-oncogenes, AP1 transcription factors working together, right? You make MDM2 mRNA that then gets translated in the cytoplasm into MDM2 proteins. And then you have other proteins that act on it, So for example, if you have strong cell survival signals that are coming in, they also drive things like our AKT, PKB pathway that result in the phosphorylation of MDM2, which is the active state of MDM2, right? So that's the state that then sequesters or binds to P53, causing the P53 protein to be recycled. So hopefully you can sort of reason this through, right? Because yes, strong mitogenic and strong cell survival signals, right, they want to carry out their functions by inhibiting P53 from doing its job of stopping the cell cycle. However, if there are, for example, DNA damage, like gross DNA damage to chromosomes, that will be detected by certain other proteins that sense the DNA is damaged, and that results in the activation of certain kinases, like these checkpoint kinases 1 and 2, that have two sort of important targets here. Right? Well, they have many targets, but the two that we're talking about on this slide are they will then do their kinase function, their phosphate transferring function, and phosphorylate P53. And we said when P53 gets phosphorylated in that transactivation region, that normally is where MDM2 binds to it, right? Then the P53 escapes the MDM2 because the phosphorylated transactivation region is now no longer the right shape for MDM2 to bind to. And then the activated P53 can now go around targeting other genes, right? Genes that would be involved, if they're expressed, their protein products would be involved in halting the cell cycle. So for example, many of the P53 targets are other CDK inhibitors or modulator proteins, right? And depending on how bad things are, right, P53 may actually trigger things like apoptosis if the cell needs to just go ahead and, take itself out of the picture by killing itself. Importantly, look, the checkpoint kinases can also function by increasing or by binding to phosphorylating MDM2, right? But these MDM2s are now phosphorylated in a different location than they were down here, right? So this phosphate position represents an activating phosphate position being attached by things like AKT signaling, activating the MDM2. Here, it's the opposite, right? These checkpoint kinases phosphorylate MDM2 here, right? And then that prevents MDM2 from interacting with P53, right? Thereby allowing more activated P53 to go on into its function. We did say that, curiously, the MDM2 gene is a target itself of P53 activity. And that's how you have this feedback loop, right? So the more P53 activity you have, there's also a tendency, right, to actually increase the amount of MDM2 you have, right, to try to balance that out. So that's how sensitive the cells are to this decision-making process of whether P53 should be allowed to interfere with the cell cycle. But it also highlights, right, how important P53 function is, that you would have such sort of a carefully balanced way of controlling it. Another example of how P53 can get activated is by this other curious protein known as ARF. Right? So ARF protein, ARF stands for alternate reading frame protein. Right? This is really kind of unusual, right? Because the ARF gene is actually the same coding region as the P16 CDK inhibitor. Right? The P16 CD4K inhibitor protein is coded for by this gene here, but they call it P14 ARF because if the mRNA gets translated with a slight shift in the reading frame, instead of getting P14, you end up producing this ARF. Right? So ARF then expression is highly responsive to transcription factor activity like E2Fs. Remember, the E2Fs are that primary group of transcription factors that want to drive the cell out of G1 into S phase, right? The E2Fs are trying to drive the G1 phase towards the restriction point and pushing the cell over the restriction point. Normally, the E2Fs are kept inactive because they are bound up by RB, right? That's how RB works. RB prevents the E2Fs from actually driving the cell cycle forward. So E2Fs also cause production of this ARF. Right? And if you have a lot of ARF that's produced, ARF sequesters MDM2 and prevents MDM2 from binding to P53. And therefore, P53 again is able to escape being degraded by MDM2 ubiquitinating it, right? So you might say this seems counterintuitive, right? Because what we're saying is E2F transcription factor activity is leading to the production of ARF, and ARF is actually releasing P53 and encouraging it to work. So again, it's a balance, right? Once again, right? The cell here has stimulatory transcription factors that are trying to drive the cell cycle forward, but part of its activity is also to encourage P53 from encourage P53 to try to keep the brakes on. This is that whole two-footed driver thing again, right? Which I think, even though it's very confusing, what it really, the take-home message is how subtle, right, and finely tuned all of these decisions are inside of our cells. So things that throw these decisions one way or the other due to certain biases, right, can really disrupt the cell's progression. So evolutionarily, that's how sensitive our cells are to being able to detect changes in instructions of whether it should proliferate or stay differentiated. So think about this. If you think about the normal level of E2F activity inside of a cell, right? So you have certain molecules of E2F that are bound up by RB, but it's not like all the E2F molecules are all bound up by RB. So you're going to have some E2F molecules at any given time that are allowed to signal, right? So if we say the normal transient level of E2F activity, right, due to proper mix signaling going on inside of the cell, you can think about it as like a pot of water on your stove, right? And it's on kind of like a low boil or a simmer. And so you have like some steam coming out, right? And just as long as you carefully control the temperature, right? Once it gets to a boil, you kind of turn the heat down a little bit and you keep it kind of simmering, right? Then you just have this steady sort of simmering pot of water with steam coming out. But if you have too much heat, right? Like you walk away from this and you forget about your pot of water, right? It might boil over. So you can think of that as sustained E2F activity. If there's too much E2F activity or too much oncogenic gene signaling going on, right, in the first place, that's doing things like turning off RB so that E2Fs are going crazy now, right? The cell can detect that. So the boiling over, as pictured in this pot here, that is suddenly the overproduction of ARF. And the overproduction of ARF really then encourages P53, hey, go to work, right? You got to stop this. You got to stop the cell cycle because we are detecting that these mitogenic signals, this oncogenic signaling happening inside of the cell is out of control, right? So let's look at this. Again, these multiple layers of stimulation on top of inhibition, which is then inhibiting another activator, right, can be very confusing, right? But if you talk your way through it, you see how these different levels of braking and accelerating all work together. So the arrival of certain types of oncogenic signals, either mitogenic signals coming from the outside that are stimulating MYC activity or RAS activity, or the introduction of some sort of viral oncogenic kind of activity through viral infection with a tumor-causing virus. So you have E1A tumor viruses. These, hopefully now, you realize all encourage, right, E2F to signal and start driving the cell forward in the cell cycle. Many of these, their pathways converge on the production of E2F protein, right? So many of these pathways just simply result in elevated E2F protein levels in the cell. But E2F is normally kept in check, right, by RB. So we would say normally RB would be here preventing the E2F from getting out of control. But as we've been learning, right, many of the roundabout ways that these oncogenic signals result in the cell cycle going forward is because they impact the function of RB, right? Either resulting in things like the E1A viral oncogenes physically interfering with RB, being able to associate with things like the E2Fs, right? Or other oncogenic signaling resulting in things like hyperphosphorylation of RB, right? Like the activity of MYC and RAS can lead to the elevated production of things like cyclin Ds and CDK4s or cyclin Es, right? That ultimately then would hyperphosphorylate RB, relieving inhibition. So what do the E2Fs do? If you have a lot of E2F activity, right, there's a whole bunch of genes that they start talking to that are involved in, oh boy, here we go, we're going over the R point, right, the restriction point heading into S phase. But as we just learned, you start getting production of ARFs, right? Elevated production of ARFs. What does that do? ARF inhibits MDM2, pulling MDM2 away from P53. So an MDM2 is no longer bothering the P53, right? And the P53, right, was originally being inhibited by the MDM2, right? So this is those double layers of inhibition. If P53 is normally inhibited by MDM2, right, by this inhibitory symbol here, and ARF inhibits the MDM2, really, you could draw this as ARF stimulates P53, right? That'd be another way to draw it. Right? You could just have an arrow coming from ARF down to the P53 and forget about this middle part if you wanted to really simplify things, right? So if the P53 is allowed to become active now, it targets genes controlling the cell cycle, so it can try to put the brakes on the cell cycle, right? So that the cell has some opportunity to see what's actually going on here, right? Or if things look really bad, right, P53 can then trigger apoptosis. So the take-home message is that too much E2F activity is a sign that RB control has been lost, right? Therefore, cells are primed to respond very quickly to this, right? If the RB control is lost, cells have evolved, we have evolved, that our cells are ready to act very quickly, right? By having P53 go ahead and arrest the cell cycle and or stimulating apoptosis. So therefore, we've talked about how RB is this very important sort of, gatekeeper tumor suppressor gene, sort of like a supervisor of many other tumor suppressor genes that makes decisions about what types of activities these other tumor suppressor genes should do. So if we call that our sort of supervising gatekeeper, then we can say ultimately P53, though, is in charge of guarding the genome, right, against irreparable damage that could be very detrimental if the damaged genome is allowed to be part now of daughter cells that are dividing. Right? So how important are things like ARF in controlling P53? So here's an experiment done in the lab where you have a strain of mice that are bred to sort of naturally express the VMYC oncogene as part of their normal tissues, right? So this is a viral oncogenic form of MYC, which doesn't respond normally or respond very well to normal MYC controls, right? And then you go ahead and you mess around with whether or not these mice still have normally functional ARF gene in them, the P14 gene, or if you have some sort of a mutant allele of the ARF, right? So in this case, they've done sort of a heterozygous knockout where one copy is still the regular ARF, the other one has been replaced by some mutated construct that's labeled with green fluorescent protein, I guess, so they can identify, what cells have this. And you can see these mice after they are born, right? The mice that still have fully functional ARF to be able to do things like properly release P53 and have P53 go ahead and stop the cell cycle. These mice, yeah, they have a compromised longevity. You can see they're dying off here just after maybe like a couple dozen weeks of life. right? Because of the fact that they have this viral MYC that's actually driving sort of this disordered regulation of the cell. But compared to those mice that have defective ARF, you can see how much more lethal that is, right? And that really pinpoints to the importance of p53 being able to properly halt the cell cycle and tell transforming cells to undergo apoptosis, right? Okay. So this is an interesting paradox. Here we have a histology slide where we're looking at a section of a tumor where on this slide they've captured tumor cells up here and the underlying stroma, right? The underlying stroma. And so, of course, these would be the epithelial cells up here. There's a basement membrane, and then below you have your connective tissue. Right? This is the supporting stromal tissue. So what you see is you see certain boundaries here where you have dysplastic or tumor-like dysplasia of the tissue. And you have up at the top here, this is really now transformed tumorous cells. And what they've done is they've stained for p53 protein levels. So this is the paradox, right? Look at how much more p53 there is in the tumor samples in the dysplastic tissue compared to the otherwise pretty normal tissue. Right? And you say, why is that a paradox? Well, wait a minute. High levels of p53 suggest what? Right? The more p53 you have, the more the cells should be able to put the brakes on the cell cycle, and the more that the cells should be undergoing apoptosis, right? So why is it that you have high levels of p53 in the tumor and transforming cells compared to the normal cells, right? That doesn't seem to make sense. Think about that for a moment. It has to do with the way that we discussed how mutations get selected for in the p53 gene that lead to the formation of tumors, right? Remember, we said that unlike most tumor suppressor genes, that when they suffer from mutations that lead to parts of them being deleted or like premature stop mutations, right? In the majority of tumor suppressor genes, those are the types of mutations that lead to then loss of function of that tumor suppressor gene, and then the cell transforms, right? So you'd expect then that levels of those proteins, you'd find them lower in the tumor cells compared to normal. But we said p53 is different, right? Because we said that point mutations in the DNA binding domain of p53 are the types of random mutations that actually favor mutation of p53, allowing for the progression of transforming cells to cancer. Why? Because again, remember, we looked at this last time, that if you have one good copy of p53 and one mutant copy of p53, let's say it has a mutation in the DNA binding domain, right? When your cells produce both versions of these, right? Maybe one came from the copy that you got from mom, one came from the copy that you got from dad that got the mutation in it. The tetramers that form out of the 16 possible tetramer conformations using this pool of blue and red p53 units, right? Only 1 out of 16 is fully functional. Right? The other 15 out of 16 are compromised because if any one of these p53 subunits, its DNA binding domain is messed up and it cannot bind to the DNA properly, then the whole tetramer cannot bind to the DNA properly. Right? So therefore, even though you have these tetramers formed, they're not functional. And importantly, right, we said that, yeah, but normally I thought the p53 gets degraded very quickly anyhow, right? Because MDM2 would ubiquitinate the p53, causing it to be degraded. Well, but where does MDM2 come from? Remember, the MDM2 gene itself is a target of active p53. So if you don't have much active p53, you actually don't produce much active MDM2 to degrade the p53, right? So as a result, levels of p53 continue to build up in transformed cells, even though this p53 is inactive, right? So that seems ironic, but that's the apparent paradox, right? That oftentimes in tumor cells, you have an abundance of p53 protein. It is just not functional. That is why, a couple lectures ago, when I showed you some early Western blotting data looking at SV40 transformed cells, remember early researchers saw that in addition to the SV40 large antigen protein levels in those virally transformed cells, they also saw that mysterious band around 53 kilodaltons, right? Which ultimately they were seeing p53, and how in transformed cells, cancerous cells, oftentimes that band was a lot darker than in normal healthy cells, right? And that's what caused a lot of confusion at first about whether p53 is an oncogene or a tumor suppressor gene. Okay, what this also suggests that I think is kind of important to think about, right, is that before you get a mutation in p53, if you're having mutations that are happening in other pathways, like tumor suppressor genes like p10 or RB, or other CDK modulators, right? Or even mutations in proto-oncogenes that are now increasing the activity of oncogenes. Just as long as you have normal wild-type p53, p53, as part of its normal job, is to go ahead and put a halt to transformation, right? I mean, that's kind of wonderful. That's how we evolved, right? That we cannot control the fact that random mutations do happen if we're exposed to things, or even through the process of cell division, mutations happen, right? And that might be in a tumor suppressor gene, that might be in an oncogene, right? But if you have p53 present, p53 will do its job of stopping the cell cycle and causing a transforming cell to undergo apoptosis, right? That suggests then that most p53 mutations that you see in an aggressive tumor, like the one that's being pictured here, probably happened later, right? Later during the whole chain of different mutations that was building up. All right. So let's talk about apoptosis then. If we say that p53 is so good at triggering apoptosis. Now, we said that apoptosis, I think you guys already know, apoptosis is basically programmed cell death, right? I made the kind of analogy last time. It's like a Hollywood movie where, they have to hit the nuclear self-destruct button, right? And then things just go bad, right? So we tend to think of apoptosis as a bad thing, but the reality is we should not, right? Because apoptosis is a normal part of tissue maintenance, and it's a normal part of tissue development, right? If we just think about apoptosis as certain cells, right, are triggered to eliminate themselves, then we can begin to appreciate how this is a crucial part of our tissues developing. So as an example, right, here we have the paw of an embryonic mouse, right? So this mouse is developing and it's starting to develop the digits of its paws, right? You can see what they've done here is they've stained these cells with some marker that stains apoptotic cells in black. So the cells that are staining in black are the cells that are currently undergoing apoptosis. Right? And what it's doing is it is removing the extra tissue between the digits of the mouse paw. Right? So this happens in us as well. Right? When we first started developing as an embryo, right, we don't have hands right away. We have like little flippers, little paddles. Right? That's sort of our primordial ancestral kind of like program of development. And then at some point, right, all the cells in between our fingers undergo apoptosis so that we can get separate fingers. If they didn't, right, we'd be like Aquaman, right? We'd have webs between our fingers. But hey, that's what bat wings are, right? I think you guys know the wings of a bat. In reality, I don't know, maybe you never thought about it, right? But what is the anatomy of a bat wing? Is it like they have an arm, a really long arm, and then they have this big flap on it? No, right? The individual ribs of the bat's wing are their fingers, right? Super skinny, long, narrow fingers, right? where the webbing between them never underwent apoptosis during development. So they end up with these big wings, right? Another example, this theme of our body oftentimes going overboard and making lots of something only to quickly destroy it, whether or not we're talking about a protein, right? Like P53 or beta-catenin, right? Beta-catenin was a good example. We make a lot of beta-catenin protein, but when we quickly destroy it because the APC GSK complex degrades it, we did those things. We take that approach because those are proteins that are needed, right, at a moment's notice if our cells detect that something's wrong and that we cannot afford to be sitting around waiting to make beta-catenin or make things like P53. But even entire cells function like this as well. Our red blood cells, which are by far the most abundant single type of cell in our body, right? I mean, it's something crazy. Like, I think it's like if your body is made up of 30 trillion cells at any given time, which obviously varies from person to person in terms of, how big or small you are. But out of those 30 trillion cells, something like 20 trillion of them are your red blood cells, right? That's crazy, right? So the rest of you, all the rest of you, that's not red blood cells, right? That's like a third of the cells that make up your body, right? So we depend upon our red blood cells, obviously, to be able to provide gases like oxygen to all of our tissues. That's why they're so important, and to be able to remove carbon dioxide from our tissues. So our blood is constantly making a lot, oh, not our blood, our bone marrow is constantly making a lot of red blood cells. But you might be surprised to know that over 95% of the erythroblasts, the developing red blood cells in your bone marrow, are simply destroyed before they turn into fine finished red blood cells. Right? So erythroblasts are sort of, as they're differentiating, becoming red blood cells, they still have a nucleus. Right? The last step is they boot their nucleus out, so they just become sacs of protein, basically, hemoglobin and other proteins, and that's your mature red blood cell that then gets released into your blood and goes around, right? And like I said, you have like 20 trillion of these floating around. But it's even more surprising to know that there are way more erythroblasts at any given moment getting very close to becoming red blood cells, but before they finally do, they are just destroyed by apoptosis, right? Why is that? Because mature red blood cells play such an important job in providing, oxygen and removal of CO2 in your body's tissues, right? That you need to be able to adjust the levels of red blood cells very quickly, depending upon changes in your environment, right? That's like if you're at sea level, which we normally, we're used to living on Oahu mainly at sea level, there's a certain percentage of oxygen in the air, right? But I think all of you know, right? If you go hiking to the top of a tall mountain, you go up Mauna Kea, or you go up to Haleakala to watch the sunrise at like 5 in the morning or whatever, right? The air is much thinner there, right? That means there's less oxygen. It doesn't mean that your body suddenly doesn't need to have as much oxygen as it's used to, right? It means your body has to quickly adapt to the fact that there's less oxygen. So how does it do that? So it simply, right, keeps many of those developing erythroblasts from undergoing apoptosis. So all of a sudden, you get a ton more red blood cells that are released into your blood, right? And you don't have to wait many hours or several days in order to do that. Right away, that starts happening. Now, of course, depending upon the activity you're doing and how quickly you got up to the top of the mountain, right, you do feel the effects of that, the low oxygen, right? You can be lightheaded, you can develop headaches, altitude sickness, whatever. But the point is over time, right, if you allow your body to catch up, then you're okay again. Even exercise, strenuous exercise, right? These days it's kind of sad. I sit on my butt a lot in my office here in the LSB building, talking to myself and my laptop, right? But if I get out and suddenly I go running around campus, right? Suddenly my oxygen demands go way up, right? So just after a 30-minute run, right, my tissues are screaming for oxygen. And so suddenly I'll have many more erythroblasts that don't undergo apoptosis that are instead allowed to become fully developed red blood cells so that I can carry more oxygen in my blood. Okay. And the third example is things like the mammary gland tissues. Right? During pregnancy, there's an enlargement of the mammary glands, the breast tissue, to be able to make a lot more glands that secrete milk. Right? And so after birth, during the first year or so of breastfeeding, right, the mammary glands, right, have a larger volume of cells in them than they normally would. And then after weaning, after the infant no longer is drinking breast milk anymore, right, naturally, those cells, they undergo apoptosis, right? They destroy themselves, right, and are essentially reabsorbed into the body. Right? So those are examples where apoptosis is not only a totally normal thing, right, but it's actually a very important thing in order for even normal tissues to function. So, not surprisingly, right, if cancer is a constant threat in our bodies, if we're talking about, several trillion nucleated cells that can all start going rogue and become any one of which start transforming, right, apoptosis is also very important then as well. And that's why I showed you that big picture of the fiery mushroom cloud. And I said apoptosis is actually a friend of the healthy, right? An enemy of cancer. So how does this work? So what we've been talking about is that any of these very distressing types of situations here that put a lot of strain on the cell that the cell may not have control over, lack of resources, damage due to radiation, uncontrolled oncogenic signaling, dramatic changes in metabolism brought on by hypoxia, things like that, or other types of defects that are blocking normal cell transcription, right? So for example, poisons and things like that might interfere with cell transcription, right? These defects are detected by P53. And then P53 may decide to halt the cell cycle to see if the cell has an opportunity to go ahead and try to repair it. But if the repairs don't happen fast enough and you have more and more P53 activity building up, ultimately that pushes the nuclear option and triggers apoptosis. So the question is, what is going on here, right? When the button is pushed, what is this process of self-destructing, like the spaceship in the sci-fi Hollywood movie, right? Okay, so let's take a look at this process of apoptosis. So here first is an overview of some of the dramatic physiological changes that take place in a cell that has pushed the nuclear option, the self-destruct. Right? So one of the first things you see is that the cell membrane of the cell loses its integrity. Right? You have the breakdown of not only the cell membrane on the outside of the cell, but also the various membranes that make up the endomembrane system on the inside, things like the nuclear envelope, the membranes that surround organelles like mitochondria, lysosomes, the endoplasmic reticulum, et cetera. And so that leads to this phenomenon that we call blebbing, right? Great word, right? But that's what it's called. So you start seeing the blebbing taking place. Inside the cell, you have the condensation of the chromosomes. So what was maybe a few moments before, the chromosomes being like big Simon noodles that were engaged before in, protein expression, gene expression, the chromosomes now condense. Right? And so you can see they form these very, very tightly condensed spots inside of the nucleus, or what's left of the nucleus. Right? Then enzymes go to work and start digesting the chromosomes. So they begin to shred up the DNA into small little pieces. The nuclei then become fragmented, right? Begins to break up. And then ultimately, right, the whole cell basically implodes. Right? Everything is breaking down. Membranes, proteins are being digested. Lipids are now, just kind of like beginning to float apart. Right? And what's left are these remnants, right? These insoluble remnants that are just called apoptotic bodies. And then these are cleaned up by immune cells like our macrophages. So our macrophages that are crawling around in the tissue looking for things like pathogenic threats like viruses and bacteria. More often, the macrophages are just doing their normal business of trying to find things that need to be cleaned up. So macrophages are like the custodial staff of our tissues, right? They go around and they clean up things like the dead remnants of apoptotic cells. So these processes, these 5 stages, seem very complex, right? There must be a multitude of different things going on in here, enzyme activities and things like that, all kinds of signaling pathways. Surprisingly, though, this happens very quickly. From step 1 to step 5, this all happens within 1 hour. So what does that mean? Clearly, this does not rely upon the cell deciding, oh, okay, I better start making, apoptotic proteins from genes that code for, apoptotic proteins that up until now were silent, right? Because that would take way longer than an hour for all of these things to be dependent upon multiple genes being turned on to make multiple different proteins that then have to work together. So again, this is an example of the cell being primed or ready all the time to undergo this process, right? So that if it needs to undergo this process and the button gets pushed, right, things happen very rapidly. That means these proteins must be present already. These different components must already be there in the cell, right, ready to activate. So when we look at a cell like this, right, you think, well, where are these proteins, right? That sounds kind of scary, right? That you have this mix of different proteins that are already in your cell ready to carry out this variety of self-destruct pathways, but they're just not active until the button gets pushed. So if we look at this, where do you think they are, right? Are they in the nucleus? Are they floating under the cytoplasm? Are they parts of certain organelles? I think some of you probably know this. So any of you, do you want to, let me see here. I can put it in the chat. You can type in the chat, or you can just speak up. Where are these critical proteins all focused on in terms of initiating P53? Anybody? Any guesses? It's one of those days when I suddenly feel like, wait a minute, maybe I never started the class and I'm just talking to myself. Okay, so it's the mitochondria, right? The mitochondria is the starting place for intrinsically activated apoptosis. What do I mean by intrinsically activated? I mean, what we've been talking about is the cell detects that there's something wrong with itself, right? And then goes ahead and activates the apoptotic machinery. That's different than extrinsically activated apoptosis. Extrinsic means there has to be some other cell, for example, like an immune cell, like a T cell, right, that comes along and communicates with this target cell and actually tells the target cell, oh, you should undergo apoptosis, right? That would be extrinsically activated apoptosis. For now, we're talking about intrinsically activated. So it starts with the mitochondria, which is really surprising. Right? And in particular, there's a component of the mitochondria that I think all of you have learned about before. Certainly, if you took Bio 275, you learned about cytochrome C, right? So cytochrome C is a molecule that's normally in the membrane spaces that separate the inner and outer parts of the mitochondria, right? And remember, cytochrome C is an essential part of our electron transport chain. Remember, as electrons are moved through big protein complexes in the inner wall of the mitochondria, that's how you end up fueling the buildup of proton gradients inside the mitochondria that are then going to power ATP synthase pumps, right, that make ATP for the cell. That's how our mitochondria make the ATP, right, is by moving electrons through the electron transport chain, and the cytochrome C is an important component of that, right? As a result, in your cell, the only place where you normally should find cytochrome C is contained inside of your mitochondria. So evolutionarily then, cytochrome C can serve a dual function. If cytochrome C is allowed to leak out of the mitochondria, right, then that might be used as a signal throughout the cytoplasm that something bad has happened, right? And that can be used as a signal to initiate apoptosis. So keeping cytochrome C in the mitochondria, right, is a way of preventing the apoptosis from being triggered. And so the way it's kept in there are certain channels on the surface of the mitochondrial membrane that are normally closed and keep the cytochrome C from leaking out. And so these channels are controlled by a group of families, a family of proteins called the BCL family of proteins. So let's look at this. So there are three sort of main groups of, or two main groups of these BCL family proteins. They're ones that keep the channels closed, right, to prevent the cytochrome C from leaking out. So those would be considered pro-survival, right? Because as long as the cytochrome C stays in the mitochondria, the intrinsic apoptotic machinery is not triggered, right? Then you have ones that are pro-apoptotic. These open the channels up and allow the cytochrome C to flow out into the cytoplasm, right, starting the chain of events that triggers apoptosis. So three of these we can look at as examples, right? Because there's a whole bunch of members, but just as long as you remember BCL2, because BCL2 is the original, I think, starting member of the family that this whole family was named after. So I believe these BH domains, refer to homology domains of the BCL2. So BCL2 is a pro-survival protein that keeps channels closed. BAX and BIM are pro-apoptotic proteins that open up those channels and let the cytochrome C out. So here you see an experiment where you have a cell over here. This is actually inside of the cell. So this is the mitochondria, right? And these are BAX proteins. BAX proteins that, if they're allowed to activate, would open up the mitochondria, right? And open up these channels and allow the cytochrome C to flow out. So if you stimulate apoptosis in these cells by treating them with this compound, staurosporine, so staurosporine, this is the structure here, sort of globally inhibits protein kinases from working. So obviously, if you suddenly shut down all the kinase functions of your cell, your cell can't survive, right? So that stimulates, you can see, the activation of these BAX protein complexes. So these BAX protein complexes go to work and they begin to open up the channels of the mitochondria, and then the cytochrome C will leak out to trigger apoptosis. How does that work? So here we have the mitochondria. Normally, the BCL2 pro-survival proteins keep the channels closed, right, keeping the cytochrome C in. But if proteins such as BAX and BIM, which are pro-apoptotic or death signals, right, open up the channels and the cytochrome C leaks out, you now have cytochrome C in the cytoplasm, right, where it doesn't belong. You have proteins known as as APAF1, which is apoptotic protease activating factor, that begin to associate with the cytochrome C. And when they bind to each other, they form what we call the apoptosome, right? The book fondly calls it the wheel of death, right? This is kind of a morbid slide, but that's what it is, right? It's like this spinning wheel here. So here's an electron micrograph of it showing that it is like this spinning wheel, right, with cytochrome Cs on the ends of these spokes of the apoptosome. The center of this wheel, right, is a catalytic site that binds and activates other proteins. What other proteins are we talking about? We are talking about caspases, right? So caspases stand for cysteine aspartyl-specific proteases. Right? And these caspases, when they are active, cleave other proteins, right, to begin to chew them up. Normally, these caspases are floating around, but in an inactive form. They are pro-caspases because they have part of the protein structure that basically covers up their catalytic domain. Right? So in order to activate them, you have to cleave off that part, revealing their catalytic activity. And so that's what the wheel of death, the apoptosome, begins to do. The first one that it associates with is pro-caspase 9, right? And turning it into active caspase 9. You can see it here now in the middle of the apoptosome. So caspase-9 is the initiator caspase. It goes to work now, and it begins to cleave other pro-caspases, removing the part of the protein that's keeping them inactive. So those include pro-caspases 3, 6, and 7, right, that then become executioner caspases 3, 6, and 7 when they're activated by caspase-9. And all of a sudden, right, these have specificity for different types of molecules inside of the cell, different types of proteins. And so all of a sudden, you get a wide range of proteolytic activity going on. So substrates like, these different proteins inside of the cell get acted on very quickly by a whole bunch of these executioner caspases that then result in that very quick succession of things like the membranes breaking down, the nuclear envelope breaking down, DNA being degraded, et cetera, et cetera. One thing I want you to keep in mind here is caspase-9 is not the same enzyme as Cas9, right? Cas9 that you may have heard of as being part of the CRISPR-Cas9 system to do gene editing, right? That is a different Cas9 protein. I think it's terrible because the names seem so similar, right? So don't get confused. Because in that case, Cas9 stands for CRISPR-associated protein 9, right? What we are talking about here, caspase-9, right, is a protein involved in apoptosis. Okay, so this slide I'm not even going to go over with you, but really what this is and it gives you examples of different types of alterations that can happen inside of the cells, right, that all sort of involve the functioning of p53 or the initiation of apoptosis. And it gets you thinking about how any of these potential genes that are involved here, if you get mutations in those genes, right, that can also contribute to things like transformation of the cell and improper survival of the transformed cell. Okay, so spend a little time just trying to look at these and thinking in your head, how would this fit in with what we were learning? Okay, you don't have to memorize it, but I think this really enriches your ability to kind of get the big picture. All right. Okay, sorry, I went really over today. So I apologize for that. Let me stop the recording.